Hi doctor, I am Liang Fei. I'll be representing Group B2 to do the presentation for Enzyme Review Video 1, which is measuring lactase enzymatic activity in the teaching lab. What is enzyme? Enzyme is a specialized type of protein that acts as biological catalyst for chemical reactions. Actions of enzymes is critical for life. This is because it provides us energy and it helps us to dispose of the waste, and it also allows organisms to function well. So, today we will be talking about enzyme lactase. Enzyme lactase catalyzes the hydrolysis of lactose into galactose and glucose. These products will later be used in the citric acid cycle, which is also known as the Krebs cycles, and oxidative phosphorylation process to generate adenosine triphosphate, ATP, which is also a form of energy. During infant development, lactose received from breast milk is one of the key sources of nutrition during early years. Lactase is highly expressed in the human digestive systems. Congenital lactase deficiency (CLD) is an autosomal recessive condition whereby mutations occurs in the lactase gene (LCT) coding for the lactase enzyme. These newborn babies will have very little lactase activity. They cannot drink any type of milk or formula contains lactose. During childhood development, lactase expression is reduced. Approximately 35% of adults worldwide have lactase persistence. Loss of lactase expressions will cause lactose intolerance that will lead to adult-type hypolactasia, ATH. They cannot break down the lactose in the gut. In the colon, the accumulated lactose is fermented by gut microbial fauna. There are released gases including hydrogen, methane, and carbon dioxide. These will promote abdominal bleeding, increase flatulence, pain, nausea, and stomach rumbling. This will also lead to loose stools. They need to reduce the lactose intakes, and instead, they will need to increase the intake of lactase supplements. Based on the video and article, a group of researchers carried out an experiment to measure the lactase enzymatic activity in the teaching lab. This method is beneficial to students as the concepts governing enzyme reactions will be demonstrated in a more proficient way. This technique is suitable for students with very little wet lab experience because they can observe and interpret the specific, measurable, and precise outcomes obtained from the experiment. Enzyme lactase was extracted from a dietary supplement tablet and observed the lactase enzymatic reaction by using a calorimetric assay based upon hydrolysis of an artificial substrate for lactase, orthonitrophenol beta-D-galactoperoxide, OMPG. The enzymatic activity was measured based on the absorbance for the reactions by subtracting the control value from the reaction value to derive the change in absorbance due to the hydrolysis of OMPG in the presence of active enzyme. Then, sodium carbonate was used to inhibit the lactase enzyme by increasing the pH, thereby terminating the reaction. However, the efficiency of initial extractions is essential to allow solubilizations of enough active enzyme for the subsequent analysis. As attested by the scientists in the video, there are a number of critical steps in this protocol. First, it is essential that the initial enzyme extraction is efficient, allowing solubilization of enough active enzyme for subsequent analysis. The other protocols are observing the calorimetric reaction, measuring enzymatic activity, and effect of temperature on the enzymatic activity. For the safety issues of this experiment, the practical was carried out according to the rules of good chemical laboratory practice. As we can see from the video, personal protective equipment including fastened laboratory coat, disposable gloves, and safety glasses are worn at all times in the laboratory. This is to protect students against accidental spills, for example, acid spills. Apart from that, we can also see that a control was prepared in the video. This is designed to minimize the effects of variables other than the independent variable. In this way, the reliability of the results can be increased, often to a comparison between control measurements and the other measurements. However, we also have a few suggestions for this video. It is shown clearly in the video that the labeling steps were being done before every process. We noticed that there would always be a step for labeling in all of the protocols. For this case, labeling can actually be done right before the experiment is carried out. This is to save time. The experiment should be planned constructively. Then, we will get to know the exact amount of apparatus and materials needed, and all this can be prepared beforehand. Apart from that, since the effect of temperature on enzyme activity involves some measurements and calculations, the set of experiments should be repeated more than once, and the average result should be obtained from all the experiments. The first reason to repeat experiments is simply to verify the results. Different science disciplines have different criteria for determining what good results are. Biological assays, for example, must be done in at least triplicate to generate acceptable data. As we all know, science is built on the assumption that published experimental protocols are repeatable. Besides that, repetition can help us to estimate the variability of the results under the same conditions and also to increase the accuracy of the outcomes. After all, the experiment to study about the effect of temperature on enzyme lactase activity can also be carried out in a simpler way. Instead of using orthonitrophenol beta-D-galactoperoxide (OMPG), we can dissolve the lactase tablets in milk of different temperatures. That will be much more convenient to be carried out in school, as the materials can be found easily from the supermarket nearby. That's all from us. Thank you, doctor. Stay safe. Take care. We miss you.